I will do this again. So now we've seen inverted, upright, and flat microscope stands. And now I'm going to talk about what is the difference between the microscope stand and the satellite equipment. So you can see here that we have a big complex system, which is one at our facility. This is the microscope stand, also called the microscope body. This is where we have the stage, where we have the eyepiece and things like this. And then everything else is what I call the satellite equipment. Okay, so that's a lot more. So the satellite equipment, so I remind you that today's goal is that you look at your microscope and you know exactly what is what and what every piece of equipment does, right? So that's what we're going to work on. So the satellite equipment can be attached to the microscope body with different types of connections. It can be cables, so I zoom in a little bit so that you can see a bit closer. It could be cables, so these, for example, are cables. It could be also optical fibers. So they look like this. They have some sort of a core. Often they have a core of metal, but sometimes it's surrounded by plastic. And these, you really don't want to bend them. And then we have hard connections like tubes or things like this. And oh my God, that is going to be very complicated. I have lots of cables and things on my microscope. You don't need to worry, because we are only interested about the equipment that is attached through hard connections to the microscope. This is what tell you what the microscope does. So we're only interested in that. We don't need to see everything else. If you wonder why I was asking you to take pictures of your microscope, it's because you're going to have to identify everything. So here is a light path tip. The sensors are mostly, or almost, I'm quite sure everybody here, they are mostly on the same side as the objective. So you can see in this, there would be an inverted microscope, and we have all this equipment here is on the same side as the objective compared to the stage, not above in this inverted microscope. So it makes it much easier. Maybe you guys can raise your hand. Maybe I can do it. So that, yes. So if you have a question, you raise your hand and I know why. So we're going to have this afternoon the workshop, Anatomy of a Microscope, where you're going to learn how to recognize all this. So the satellite equipment, which is attached with hard connection to the microscope body, they can be cameras, scanners, and a few more. So I want to remind you that the world of microscopy is divided into the microscope without a camera at all, and they cannot do any imaging. We don't care about those in this course, okay? We're only concerned with microscope where we can actually acquire images. But we can have microscope without cameras that are can do imaging, and these are single point confocals. So that's what we call confocal. Otherwise, we can have microscopes with a camera, and these are all the other microscopes, okay? If there is no scanner on this microscope with a camera, if there's no scanner, it's a wide field system. If there's a scanner, it's a multi-point confocal. So single point confocal, multi-point confocal. This is called also a spinning disk. Most people call multi-point confocal spinning disk. To tell you the truth, I have invented this for this course. So, and there's a few more things, but mostly this is what it is. So it makes it quite simple, because it means that during the anatomy of the microscope workshop, we will learn how to recognize a camera, and we will learn how to recognize a scanner. And once you can do this, then you can just look at any system and you can identify if it's a wide field, a single point confocal, or a multi-point confocal. I thought I had something here, but I don't have. But never mind, I will define that later. It's okay. So now that we have seen the difference between what the microscope stand or microscope body is and the satellite equipment, I'm going to talk about the difference between transmitted light and fluorescence. So in hide the controls. So in transmitted light microscopy, as I said before, one of the major techniques is called bright field. And bright field looks like this. An image acquired in bright field looks like this. We have a dark object on a bright background. You see, the background is almost white, and then we have an object that is darker. And these are beautiful images that Waldo Nell was kind to let me use. So the light goes through the sample in transmitted light microscopy. So the sample must be transparent and very thin, right? So this is mostly what is used at the hospital as well. So this transmitted light microscopy is available on wide field microscope and confocal microscope. So again, wide field microscope, do you remember? This means we have defocused illumination, and we're going to talk about this. Wide illumination. So a transmitted light microscopy is a low contrast techniques. It's a little bit like you're looking at the sky during the day, especially today. It's a very white sky. So we look at the sky during the day and we try to see the stars. We do not see them. Why? They are there, but there is a low contrast. So it's a low contrast technique. Also, in transmitted light microscopy, there is no optical sectioning. It doesn't matter if you acquire a transmitted light image on a wide field system or on a confocal system. Anyway, the image has no optical sectioning. Okay? So the contrast can then be enhanced to help. And this, we can enhance it two different ways. We can enhance with optical elements. And here we have differential interference contrast. This is one type of transmitted light microscopy. So bright field is one, DIC is another one. And then phase contrast, we have different elements again in the light path. And this is what the image looks like. And this is a way to increase the contrast. But we have, in these images, we have a high optical resolution, but we have a low contrast. And also we have no molecular specificity. So nothing is labeled in these images. So they have no molecular specificity. So the contrast can be enhanced with optical elements. It can also be enhanced with colored dyes. Like for example, here we have eosine hematoxylin in this beautiful image. And unfortunately, there are very few colors available. So we can have blue, pink, and brown, more or less. And also this requires a color camera. So we will talk about the different black and white and color camera later. So what are the applications for transmitted light microscopy? It's excellent to put fluorescence in the context of an unlabeled tissue or cell, because when you look at fluorescence, you're only looking at what is labeled or autofluorescence, but you don't see the whole thing. Whatever is not fluorescent, you cannot see it. So it's a great way to have a context to use also the transmitted light microscopy image. It's also a much cheaper technique, and so it's used a lot in the clinic. And by the way, this is label-free microscopy. This has not been labeled, whereas this one has been labeled. There's also another technique of transmitted light microscopy. It's called dark field microscopy, but this is where the object appears bright on a dark background. But this is pretty much used, I mean, it's used a lot in material imaging more than in our field. So I will not talk about this. And here. So you can see here in these images of transmitted, yes. I have a little shrug. You can see here in these images of transmitted light microscopy that were analyzed with AI, that it is very nice to have this possibility to track the cells in here with these unlabeled cells. The nucleus has been tagged with AI that can recognize these, or in here as well. So we can do really fancy stuff with transmitted light microscopy, thanks to AI nowadays. So here we go back to our light path. What is the light path for transmitted light microscopy? So remember, there is light source and sensor, and in between we have optical elements. So on this inverted system, inverted microscope stand, the light path is, as always, from the sample to the sensor, and it goes through the objective. So here we go, we have the stage with the sample, then we have the objectives here in the inverted system, and here we go to the sensor. So either we have a camera here or we have the eyes. But where does the light come from? So where is the excitation? What is the excitation light path? So the light comes from the light source and it goes to the sample. Because it is something that always true in light microscopy, because we are talking about transmitted light, the light goes through the sample. And the sample absorbs some of that light, and the rest of the light, which is not absorbed, is going on the other side of the sample. That's why the sample has to be thin. The light goes through. That's why it's called transmitted light microscopy, right? So for that reason, the light source must be on the opposite side compared to the sensor. So the light source is here on this microscope, and then it goes through the condenser, goes to the sample on the stage, and then we have the emission light path. So this is the excitation light path. This is the emission light path. The light goes through the sample. So the light source and the sensors are always opposite, on opposite sides compared to the sample for transmitted light microscopy. And the light goes through the condenser. Okay, so this is what is always true for all microscopes for transmitted light microscopy. And in most cases, the light source is a halogen lamp or a white light LED. So you will have to tell me what is the light source for your transmitted light when you image transmitted light on your microscope. This is one of the assignments. And the light source is opposite the objectives for most of the time. There's only one exception, and you maybe have noticed that it's always the same exception. When we image transmitted light with a single point confocal, it doesn't work that way. So we don't have the light source opposite the sensor. In that case, and why is that? It's because we use the lasers as light source in a point confocal, a single point confocal. And so it's the same lasers that are used for fluorescence and for transmitted light. And so the light source is here at the bottom. And then it goes through the objective, it goes through the sample. And because we're transmitting light, we have to add an extra sensor here to be able to collect the light that has gone through the sample. Okay, so this is the way it works for imaging on a single point confocal when you image transmitted light. This is the way it works. But anyway, in transmission, when we image transmitted light, the light always goes through the sample. That's the principle. So here you have the two light paths that are aligned. The one where we are looking at fluorescence, or we're looking with a camera here. And then when we are imaging on a single point confocal, it's the opposite. We have the light source here and the sensor here. Okay, so let's have a little poll now. I would like you to raise your hand if you know if your microscope or let's say raise your hand if your microscope can acquire transmitted light images. Okay, pop, pop, pop. Right. So we have a couple of people who maybe do not know. It's possible as well that you've never acquired a transmitted light images and you do not know. But after this course, you will know because you will have to acquire a transmitted light image with your microscope. So have you ever, let's bring down your hands, everyone. And then you tell me, have you ever acquired a transmitted light image on your microscope? Yes? So quite a lot of people have acquired transmitted light images. Transmitted light images are fantastic to put the fluorescence in the context of the tissue or the cells. So it is very useful. Okay, excellent. So now we're going to have a little quiz again. So you go back to Canvas, and we're going to have this quiz called Light Path for Transmitted Light. I spoke about transmitted light microscopy, and now we're going to, let me see, lower. Now we're going to talk about fluorescence. So fluorescence is very different from transmitted light microscopy because now we have a bright object on a dark background. So in fluorescence, we have two types of lights involved, right? So we have two light sources. The excitation light is very, very strong, and it can come from different light sources. It can come from a mercury lamp, it could come from LEDs, or it could come from a laser, and it goes to the sample. But the emission light is very weak compared to the excitation light, and it comes from the excited sample, and then it goes towards the sensor. Okay, so here we have the excitation light path, and here we have the emission light path. Fluorescence is available on wide field confocals and light sheet systems. And it's a very high contrast technique. So we have, yes, it's a bit like looking at the stars, this time during the night, so you can see them. And the contrast can be further enhanced anyway using fluorophores. The advantage is that we have many different types of colors that mostly we can use black and white sensors, and we'll see later, black and white sensors are more sensitive than colored sensors, so it's very good to be able to use them. And also but that requires us to separate the different colors. And we know now how to do this. We can use filters to separate the different colors. So the applications for fluorescence microscopy are we have a very high molecular specificity. So we can image, for example, we can use, but we also have a high resolution and a high contrast. We have all this together. So we can label molecules with antibodies that only label a protein when it is phosphorylated, very high molecular specificity. We have a high resolution and a high contrast, all this together. And I remind you that for transmitted light, we had a low contrast. We can have for transmitted light either a high molecular specificity by using this type of antibody or a high resolution. And we will see this why a bit later. So what is the light path for fluorescence? The light path is the same type of things. It goes from the light source to the sensor, and there we have the optical elements in between. So on any microscope, for the light path for fluorescence, we have the light source that delivers the excitation light. And so it can be mercury, LED, or lasers. That's true for any microscope. And then the excitation light is delivered through the objective. So the excitation for fluorescence always goes through the objective. So the light source is clearly always on the same side as the objective compared to the stage. And the excited sample then emits light in all directions. And so in most cases, for most microscopes, only the emission light that goes back into the objective is detected or viewed. Okay, so everything else that goes on the other side of the stage is not detected. So that means that in fluorescence microscopy, both the excitation and the emission light go through the objective, and then they go to the sensor. Okay, this means that the other side of the microscope is completely not used at all. Okay, if you go to your microscope next time and you're imaging, you could, if it's an inverted system, you could flip the top of the microscope open. The image is not going to change because on an inverted system like this, the top of the microscope is completely not used in fluorescence microscopy. It's only used in transmitted light microscopy. So in most cases, the excitation and the emission light go through the objective for fluorescence. We cannot use lasers when we look in the eyepiece. So when we are looking in the eyepiece, we need another light source on our systems. We will never use lasers because it's dangerous for our eyes. In that case, when we look in the eyepiece, we'll always use a fluorescence lamp, like a mercury lamp or LEDs. So here is a little summary of everything I've said so far. So we can have transmission microscopy and fluorescence microscopy. And we can have, for all systems, we can have inverted or upright microscopes. So I'm going to show you all this. Here is an inverted microscope for transmission microscopy. We have a little fish as a sample, and this is the objective. And then it's the same thing for a fluorescence microscopy if the microscope is inverted. And here is if it's upright, the objectives are above the sample. And then the condenser is always on the other side of the sample compared to the objective. So we have the condenser here. This is for transmission microscopy. In fluorescence, we don't use the condenser. So, okay, no need for a condenser. It's maybe if it's there, but we don't use it. The sensors are always on the same side as the objective. So here we have, I represent it like this, but it can be a camera or a detector or the eyes. It's always on the same side as the objectives. But when we image transmitted light, the light source is on the other side of the sample because it has to go through. So here we have the light source here on this inverted system. And then on this upright system, the light source is below because the sensor is up there. And in fluorescence, the light source is on the same size as the objective. So this is quite different. That's why we don't use the other side of the microscope. So we have the light source and the sensor in the same side. Both the excitation and the emission go through the objective. So there's only one exception, and it's always the same exception. It's for you guys who image with a single point confocal and image transmitted light. It doesn't work this way. And that is because we use lasers. So here is the way it works. For an inverted system, we use the same light source as for fluorescence, but we add an extra detector. And it's the same thing for upright, just flipped. So this is always the one that is different from the others, is when we have a single point confocal with transmitted light. But most of you are in this situation. Either you have a camera-based system inverted, or you have a single point confocal inverted. And this is for transmitted light, and this is for fluorescence. So this is what summarizes pretty much the excitation and the emission light path of your systems in the course. So here are a few conclusions quickly, and I am looking at my time. I think I'm doing okay. Yes. So do not confuse wide field and bright field. They are different. And microscope stands can be upright, inverted, or they can have a flat architecture. And one of the major differences between fluorescence and transmitted light microscopy is the contrast. Transmitted light microscopy is a low contrast method. Fluorescence is a high contrast method. And that leads to a world of difference between these two techniques. When we look at the light path, it always starts with the light source and it ends with the sensor. And the sample is part of the light path. The sample and all sorts of other optical elements are part of the light path. But you need to remember that the microscope manufacturer assumes something from your sample. And the closer you are to this assumption, and the better for the images. We will talk about all this in the sample preparation lecture. So in transmitted light microscopy, the light always goes through the sample. And then in fluorescence, both the excitation and the emission light go through the objective. And the light path for transmitted light microscopy is quite different between single point confocals and the other types of microscopes. So this is a little bit of an exception we will go through. Right, so now I'm finished for this little lecture, and what we're going to do now is we're going to go to the workshop Zoom. We will do a few quizzes, then we'll have a little break, and then we'll come back to this Zoom for the next lecture, which is at 10:30.